First off, so thank you so much for the opportunity to present on the Quagga mussel response in the Snake River in Idaho last fall. And it'll be very much focused on a Snake River response, not so much the other parts of the program, but just keep that in mind that there's everything from prevention, watercraft inspection, to monitoring, to active control programs, to education outreach that's going on in the state of Idaho, many of which that are working with local cooperators, including tribes, of course, as we're rolling out this program. But just for the interest of time, we'll keep on to the mid-snake. Just give you an idea. This talk can generally take about an hour to cover the whole story. So I'll try to really condense it, hit on the high points, really give the group an idea of what it looked like on the ground. I know we had many folks that were out there in the mid-snake as observers during the treatment or during the response, which was great to see. Many of them were maybe on this call even. So, but just to give everybody an idea of what it looks like on the ground. And really the story starts with our annual monitoring program. So we've been doing monitoring as well as watercraft inspection for about 15 years to the state of Idaho, long before I was even a part of the program. You know, this has been going on, and the monitoring very much focuses on a couple of different life stages, whether that's looking for veligers under a plankton tow sample, looking under cross-polarized light microscopy, or looking for adults in various water bodies throughout the state, whether it's doing shoreline walks during drawdowns, ponar grabs, looking at artificial substrate, things like that, those type of techniques, looking for adult mussels on substrate. You can see we're all over the state of Idaho. So clear up from the upper Snake River Basin down through the mid-snake, the lower Snake, other drainages, you know, the salmon, the Clearwater, the Coeur d'Alenes, obviously Pend Oreille and her tributaries, and the Kootenays. And so really every water body is classified throughout the state as far as, you know, if they're critical, high, moderate, low, and just the frequency that we're out there sampling. But this is really where that story starts. And, you know, going through the 2023 monitoring season, it really, on September 18th is when we had enough information that's required by our Idaho Rapid Response Plan to initiate the response. And what that looks like is multiple sampling events, so not just one plankton tow sample on one date. So we're really looking for multiple sampling events, preferably multiple samples, and multiple specimens per sample container per plankton tow. And so that's when we're actually looking for the body underneath a microscope. And then we're going to take a subsample from the water or from the specimen itself to send it off to qPCR to get genetic verification. And on September 18th, that's essentially when all that information came back to the department. I remember that date very well. It came in the morning, and by the afternoon, we were sitting in the governor's office rallying the troops about what we're going to do about quagga mussels in the mid-snake. And fortunately, Idaho, as well as other Pacific Northwest states and our Pacific Northwest group as a whole, we have rapid response plans in place. This is something that Stephen Phillips with Pacific States Marine Fisheries Commission works on, as well as other states, coordinating a response, methodical, strategic on, you know, if you get quagga mussels, what do you do? And this diagram kind of shows that, and I'll go through the steps, but really that response and the meeting with the governor's office and the stakeholders is really what initiated that response. And the Idaho Rapid Response Plan is simply just a framework. There's not very many details because frankly, the details come from the water body specifically. A response in the mid-snake is going to look drastically different from a response in the Pend Oreille or Bear Lake or Redfish Lake because characteristics, water usage, volume, it all changes. So again, the Idaho Rapid Response Plan is simply just a framework. Within that, you've got these main pillars, you know, notifying impacted entities. That's the whole communication piece, is how do we get the message out to the public? And I'll go through that. Then we implement containment measures, you know, whatever that looks like, whether it's a closure, decontamination, things like that. And I'll talk specifically about the mid-snake for containment. conducting delineation surveys, so trying to find the extent of the problem, and then starting to explore the treatment options. So really, this was the punch list that we went through on September 18th. And essentially, each one of these pillars had its own page. And we just started writing in notes and thoughts about specifics to the mid-snake and what we were going to do for communication, containment, delineating, and treatment, and really getting those first thoughts on a piece of paper. You know, the first part is really that communication piece. So how do we effectively get messaging points out to the public, out to the stakeholders, to industry, talking about the response in quagga mussels and the mid-snake, and really what that means to the state of Idaho. So we quickly realized we needed to establish a communications team, not just within the Department of Ag, but really multi-agency. So Idaho Fish and Game, DEQ, Water Resources, really that whole picture. That way we can get all those public information officers all on the same sheet of messaging points and getting consistent, clear, concise messaging out. It's all factual, it's to date. We quickly realized that the communication needed to be fluid, right? Just like the scenario that we're in, it needed to have the ability to change as the response progressed. So the first few media releases very much focused on the detection itself. Then we started focusing on the containment and the closures. And then we need to start talking about treatment and having very concise messaging points about that specifically. And we had multiple media releases. They kind of look something like this. We did press briefings and press conferences and worked with local media outlets to get the messaging out, create a web page. We had an invasive species hotline. So we had to get all of our administrative staff up to speed on the response, and that way they know the talking points and how to answer questions from the public. And again, really speaking to that, adjusting the message as the response progressed. I should really take a step back, and when we're talking about the four main pillars, one of the key decisions we made early on is to run all those tracks at the same time. And that's because of the response the detection coming in late September 18th. We didn't really have a whole lot of time before the end of the field season, which typically ends around October, November, is when fall and winter starts setting in, and we're gonna have some environmental factors that's gonna kick us out of the river. So we had to consolidate all this, and so we had to basically run all these tracks at the same time. And the next one being implement the containment measures. So we had to quickly identify the authority to implement a closure, literally closing off boat ramp accesses, barricading, chaining, putting gates up. We identified that within the Idaho Plant Protection Act and the Idaho Invasive Species Act, as well as the IDAPA rules, the administrative rules that accompany those acts. We set up mandatory decontamination for exiting watercraft. And then we also set up a big campaign and 2 decontamination stations in the city of Twin Falls at 2 locations that the public really knew well. One at the visitor center and one at the County West building, which is the old county hospital. Basically places that we could outline in a media release and say, yep, the public isn't going to know exactly where that location is at. We just simply need to post the hours of operation and the expectations about what the interaction is going to look like. In the first weekend, I can say that we had great turnout for the decontamination sites, and that really progressed as the response continued. A lot of boating public was coming to us, following the standard operating procedures or the protocols: hot water, 140 degrees, 10-second contact time, really free of charge. No questions asked, just come to us, get your boat decontaminated, and off you go. This is a little bit about what that looks like on the ground. Decontamination mat, everything from canoes, kayaks, paddleboards, duck decoys, to john boats, fishing boats, clear up to larger scale wakeboarding boats as well. Life jackets, anchors, everything that comes in contact with the water, we needed to decontaminate. This is another picture. All right, now we're shifting gears to the third pillar. This is the conducting delimitation surveys. We do this with other agriculture-related pests. It's basically an effort to find the extent of the problem. So with quagga mussels and the mid-snake, the strategy was to start upstream with clean, brand new nets, taking tows, plankton tows across the river current, across the channel. So you take one tow, two, three tows across the river channel. That's one sample. Travel directly downstream and then do it again. And so that's sample number 2, across the river channel, directly downstream, do it again. And we essentially did 100, pre-treatment delineation surveys within that mid-snake region, using that strategy, tow, downstream tow. And really it was a strategy you're going to have 0 veligers, non-detects upstream of the population. And then at some point, you're going to encounter veligers, preferably a plume of veligers or an area of increased veliger production, which would indicate an adult colony of mussels below, somewhere producing the veligers. And so that was the strategy. The very focused delimit was between Twin Falls and Centennial Park, or the wastewater treatment plant. At the end, we stretched that delimit sampling all the way upstream past American Falls. This was to try to answer the question of, what's going on upstream? Do we have a problem upstream? Fortunately, we didn't detect any quagga mussel veligers upstream of the Twin Falls Dam. And then we extended the delimit samples downstream past CJ Strike into the Brownlee area. And this was to help answer the question of, what's downstream movement look like at that time. And this is basically the chart showing the map showing the delimit sampling points. So these are the sample locations, not the quagga mussel veligers, so just the sample locations. You can see we're in Milner, around the Burley area. Very fortunate to date, we have not found any veligers in the Burley area, in Milner. And why I say that is because the Mid-Snake and Burley and Milner Reservoir, there's extensive water diversion going out of Milner. Essentially, the entire Snake River at times is diverted either north to the Northside Canal Company or south to the Twin Falls Canal Company. And there's miles and miles of laterals and irrigation canals and reservoirs that feed the Magic Valley, basically one of the main hearts of Idaho ag. And we haven't found any sign of quagga mussels upstream of Twin Falls Reservoir. And so there's no problem that we know of in Milner. And then you can see right there around the city of Twin Falls, Kimberly, Buell area, that's very focused delimit. And then you can see downstream towards Hagerman and CJ Shrike area. Now, really focusing in, again, these are sampling points. You can see the delimit samples between Twin Falls, Shoshone Falls, Pillar, Centennial Park. And then this is the information that came back from the microscopy lab showing where the veligers are. And the yellows are somewhere between 1 and 5 veligers. Orange is 6 to, I think, 15. And then the big red one, I think, was around 30 veligers per sample container. So that's our plume. Just directly downstream of Shoshone Falls is our plume of veliger production. And that's when we deployed the divers, and we actually found one quagga mussel underneath the river, attached to some substrate about 20 feet from the bottom of the river. So you can see all of our delimit sampling points there. The white is non-detect. So just keep that in mind. Those are samples that are taken, but no veligers were found. So this was all gearing up towards a treatment plan, which was, of course, another big pillar of the project. This team was working at the same time. The communications, the containment, and the delimit sampling were taking place. There was a team very much focused on treatment, exploring treatment options, products, labels, permitting, rates, application techniques, things like that. And one of the products they selected was Natrix. It's a chelated copper product. It's manufactured by CPRO. It's main things that played into the selection of this product was it's labeled for the specific site use for the organism. It's already registered in the state of Idaho for use in Idaho. The product manufacturing was available, so they were able to ramp up and provide the quantity that was needed, with the target rate being 1 part per million for 2 96-hour treatment blocks. You really need that extended duration, because the way it's going to work is the quagga mussel is going to open up, they're going to taste a little bit of copper, and then they're going to close right back up. And so they're going to try to hunker down as long as possible. And that's really why you need that 96 hours at that 1 part per million to wait for that quagga mussel to open back up and feed and take in that product to make it effective. You really lose efficacy when you drop down below the targeted rate or below that 96-hour duration. But more specific site data was needed to really ramp up and produce the treatment plan, depth profiles and bathymetry, flow rates, so CFS, and really locking in those CFS to being really consistent, water chemistry such as pH and calcium, and really the water flow characteristics of that section of river being the inflows, the outflows, and all the springs, upwellings to be concerned about. This is very much the treatment polygons. These were directly derived from the pre-treatment delineation surveys. So you can see the area that we found the adult quagga mussel, the area that we found the plume directly below Shoshone Falls. But we had 3 treatment polygons: Twin Falls, Shoshone Falls, and then Pillar to Centennial Park. We had to do some checks with EPA, US EPA on the label, as well as US Fish and Wildlife Service on ESA listed species, and that type of a consultation. And so we were able to get a quick approval on both of those. One of the key things that played with the treatment was an on-the-ground meeting on Pillar Falls with all the water managers associated with the mid-snake. So all the stakeholders that had a stake in the game, a straw coming out of the river, whether it's the federal government being BOR, USGS, BLM, whether it's other state agencies such as Idaho Fish and Game, Water Resources, DEQ, Emergency Management, and then a very local stakeholders being county commissioners from Jerome and Twin Falls County, the city of Twin Falls, and the mayor herself was there on Pillar Falls. And so really this group, very specific to the mid-snake, came together pre-treatment. We all stood out on Pillar Falls and said, here's the treatment plan, here's the scope of work, all the treatment polygons, the product, the label, the permitted use. Here's the game plan. This is your chance to ask questions, voice concerns, say yes, say no, we want to hear it. That way we're all on the same page. And coming out of this manager meeting, we were all very much in favor of performing the treatment.
Nick, just about
and I know I just got. Yep, perfect. I'll wrap up just about 3 minutes. The treatment notifications, we had several different efforts going on: pre-treatment, signage, message boards, paid advertisements, coordinating with the HOAs, the private docks along the river, door knockers, literally going door to door within the treatment area and 16 miles downstream, both sides of the river. And then on Sunday, prior to the treatment, we conducted a town hall meeting to go through the treatment plan, both virtually and in person. A lot of questions are around downstream movement of copper. We modeled and projected 16 miles of downstream movement before the copper would be diluted, taken up by plants and algae, broken down. And so really we wanted the copper to move down a little bit to help address the concern of downstream movement of villagers, but we needed to project and model how far it's going to move down. And this 16 miles ended up being just about right. So during the treatment, we were measuring copper downstream, and we stayed well within that range. As a contingency, we did purchase some activated charcoal wattles in case the model was incorrect. These wattles you can deploy in sensitive areas to help deactivate the copper from the water column and bring that copper down. We had these on site, but we ended up not needing to use them because, again, the model worked as we were expecting. We had to do a bunch of staging of equipment, so flying in survey boats, moving totes. All these had a different site location, whether we were going to move it on a truck, on a boat, with a helicopter. We used natural features within the waterfalls to place totes. And we used the hydroelectric facilities at Twin Falls and Shoshone Falls as placements for a drip into the intakes. This was really effective because we could really keep consistent CFS flow and really lock that in and have a really good mixing zone. And it was really secure for our equipment. The treatment itself was conducted, it was started on October 3rd, 2 96-hour treatment blocks or 192-hour treatment. It ended on October 13th. We utilized just under 47,000 gallons of product to achieve that 1 part per million. And then copper was actively monitored until it went back below baseline levels on October. Again, this is what that looks like at the intakes within treatment boats going over the falls. Again, 96 hours contiguous, so it's around the clock. And I'm coming up on time, so I'll just show you a few more pictures. So the treatment did impact the adult mussel. We had 100% mortality of that individual within 48 hours of the first treatment block. Preliminary results look really good for veliger samples. And the reason why I say that is because we've got a long ways to go. So we've got 5 years of extensive monitoring in the mid-snake before we could ever call this any type of eradication or success. So we're very much in a marathon now. And then we're shifting from a containment to now, or a prevention to now a prevention containment state. And so there's going to be mandatory decontamination at Centennial and Shoshone Park, extensive monitoring statewide, expanded outreach and education programs. And this is my last slide. I'll just wrap up by saying I get the opportunity to tell this story, but it's not really my story to tell. It's not even really one of the agency. This was really a interagency, multi-agency coordinated effort that we all identified a problem and very quickly addressed it. Thank you so much. I know that's a lot in a little bit of time, but that'll give you an idea of what it looks like on the ground.